Hey, so here's some words you've probably heard before. DNA, gene, chromosome, genome. Maybe you've even heard the word chromatin. Today we're going to make sure we understand these words and how they relate to each other because it's actually kind of easy to get confused with all of these terms that have to do with DNA. We've already learned what DNA is, deoxyribonucleic acid. It's a double helix that consists of two strands. Each strand is a polynucleotide, and the strands run anti-parallel to one another. And they're held together in the middle by hydrogen bonds between the base pairs. But these other words refer to DNA as well, and we need to know what they mean. We're going to start by thinking about you. You have a genome. A genome is all of the DNA, all of the genetic information in a cell or organism. Let's look at a typical human cell. Most of the DNA is contained inside the nucleus in humans, but we do have one source of extranuclear DNA, which is the mitochondrion. Your cells generally have lots of mitochondria, and each one has its own circular molecules of DNA. Each one is 16,569 base pairs long and contains 37 genes. 13 of these genes code for polypeptides, shown here in green, and 24 code for non-coding RNAs, shown here in orange and yellow. The reason that mitochondria have their own DNA is because mitochondria are the result of an ancient endosymbiosis. They began as free-living bacteria, but became engulfed by an ancestor of eukaryotic cells. Over time, they were transformed into organelles, and now they can't live without their host cells, and their host cells can't live without them because they have the function of producing ATP, cellular respiration. They still divide like bacteria, though, and their DNA is strikingly similar to some species of bacteria today, such as those in the Rickettsia genus. But by far, the bulk of the human genome resides within the nucleus of the cell. Most humans have 46 chromosomes in each cell. These are paired, so 23 pairs of chromosomes equals 46 total. This is a picture of the chromosomes. Notice that they're not all the same size. Chromosome 1 is huge, for example, while chromosome 21, chromosome 22, and the Y chromosome are comparatively tiny. One of these pairs is known as the sex chromosomes. Most humans have either two X chromosomes or one X and one Y chromosome. Each of these chromosomes is an incredibly long double-stranded molecule of DNA. Altogether, the 46 chromosomes contain about 6 billion base pairs. By the way, you'll often see the size of the human genome quoted as 3 billion base pairs. That figure is correct too. It's actually the more usual way to cite it, and it refers to the base pairs present in 23 chromosomes, or the haploid number of humans. Remember, 23 pairs equals 46 chromosomes, and 3 billion times 2 is 6 billion. Now, to be honest, chromosome is a bit of a sloppy word. Some biologists use it to refer to the DNA molecules no matter what's going on in the cell, but others are more specific, and they use it to refer to the DNA molecules only when they're tightly condensed because the cell is dividing. When that happens, chromosomes can actually be visible under a microscope, and in fact, that's where the word chromosome first came from. It means colored body, and scientists were able to apply colored dyes to cells and actually visualize the chromosomes because they were stained with these dyes. But I'm going to use the word in the broader sense. So I'll call these DNA molecules chromosomes, whether they're visible or not, and whether the cells are dividing or not. And in a eukaryote like you, DNA is associated with proteins. The main proteins are called histones, and they help to organize and package the DNA into a manageable form. This is necessary because you have a lot of DNA in each cell. If you were to string it end to end, you would have a string of DNA that was really, really thin, but a bit over 2 meters long. And it has to fit inside a nucleus that is probably around 6 to 10 micrometers in diameter. This complex of DNA and protein is called chromatin. When the cell is dividing, it's extremely tightly packaged, similar to what you're seeing in these chromosome structures on the left. These are labeled with fluorescent dyes so that we can see them. Chromosome, colored body. When the cell isn't dividing, though, the chromatin is still packaged, but it is more relaxed, like on the right. Here again, the chromatin is labeled with fluorescent dyes so that we can see them. Notice that the color is more diffuse and spread out, and we don't have those nice tight shapes that we can see as distinct things. It's all kind of diffuse. This is what the chromatin is normally like when the cell is not dividing. I like to think about this as a bowl of spaghetti, and there's a reason that we see these two forms of chromatin. Structure is function. If the cell's not dividing, if it's just living and metabolizing, well, we have to be transcribing genes all the time. We need to copy the information that's contained in those genes into molecules of RNA so that we can make polypeptides, proteins, which are completely necessary for maintaining life. We have to do that all the time. And we can't do it if the chromosomes are so tightly packaged that the proteins that function in transcription don't have access to the strands of DNA. On the other hand, have you ever tried to divide a bowl of spaghetti exactly in half? Seems impossible. And for the cell, it would be pretty difficult because if the cell is going to divide, it first needs to copy its chromosomes and then divide them evenly between the two daughter cells. So each has an identical copy of the genetic information. And that's not going to happen correctly if our DNA is spread out in this bowl of spaghetti, looser configuration. We need to really tightly package it so that each daughter cell comes out of cell division with a whole genome. Okay, so we've covered chromatin and chromosomes. Let's talk briefly now about genes. We've discussed genes before, so you should have a basic idea of what they are. They're segments of DNA that contain the information to make a functional product, either a polypeptide, think about proteins, or a non-coding functional RNA molecule. How does that relate to chromosomes and genomes? Okay, so every chromosome has many genes on it. Humans have about 20,000 protein coding genes, give or take, maybe a little less. We're not entirely sure how many genes there are that specify non-coding RNA molecules because it's hard to predict. But let's just think about the protein coding genes. Okay, approximately 20,000 genes divided over 23 chromosomes. And it's not an equal division. Remember that the chromosomes are different sizes. Chromosome 1 seems to have just over 2,000 protein coding genes, while the Y chromosome has approximately 70. It's really different. We'll learn more about genes later, but for now, let's just emphasize that the information in a gene is contained in the sequence of bases. For example, let's take the enzyme phenylalanine hydroxylase that we've met before. This is the enzyme that catalyzes the production of tyrosine from phenylalanine. The enzyme is encoded by a gene, PAH. This is the first several bases of that gene, ACT, CTC, TTC, TCC, TCC, CTA. Of course, it's longer than this. There are 3,759 nucleotides that code for 452 amino acids. These amino acids are folded to make a subunit of the functional protein, which has either 2 or 4 identical polypeptides. Okay, so to recap, your genome is all of your DNA. Humans generally have 23 pairs of chromosomes for a total of 46 in each cell. Well, most cells anyway. Each chromosome has many genes. Each gene contains the information to make a functional molecule, either a protein or a subunit of a protein or a functional RNA. All right, hope that helps. Bye.